Hello everyone, and in this video we're going to be talking about how does Johnson & Johnson's new COVID-19 vaccine work, and we're also going to be discussing how it relates to the Moderna and Pfizer or BioNTech's current standing for their vaccines. And so the very first thing we're going to be looking at is a coronavirus, and we know that this coronavirus is coated with these proteins on the outside of its surface, these red things. These are called spike proteins. These spike proteins are what allow the coronavirus to actually stick onto cells before it infects them, and it's involved in this fusion event that occurs in which that coronavirus is able to actually put its genetic payload into a cell so that that cell becomes an infected host that can go on to create a bunch of viral progeny. So basically the key takeaway from this picture of the coronavirus is that it's got a bunch of spike proteins on its surface, these red things, and these are the things of interest that these vaccines are targeting, whether it's Moderna, Pfizer, BioNTech, or in this case we're looking at the Johnson & Johnson offering. And so the specific thing that Johnson & Johnson is doing is they're taking this thing called an adenovirus, and inside of this adenovirus you've got this protein capsid, which is this outer shell, and then inside of this shell, just like the actual coronavirus, you've got this genetic payload. But unlike the coronavirus and unlike the Moderna or Pfizer vaccines, instead of mRNA, we're making use of DNA. And so by making use of DNA, Johnson & Johnson has a leg up on the Pfizer and Moderna offerings because DNA is inherently a much more stable molecule than RNA. And the reason for that is that DNA is double-stranded and it has more hydrogen bonding inside of it to help it be more thermally stable. And the reason that this matters to us at the end of the day is that when it comes time to actually distribute and vaccinate people in the real world, it's a lot easier to store and transport this stuff logistically. So by having a more stable molecule to begin with, we don't need to worry as much about making sure that we keep this stuff at like -70 and actually store it. In addition to that, because DNA is so much more stable than RNA, you only need to have one injection of the Johnson & Johnson vaccine in order to be immunized. Relative to Moderna and Pfizer, you need to have those two injections, those multiple jabs, about 4 weeks apart. And so the reason for that is because the mRNA that is inside of those vaccines is so unstable, it's readily degraded inside of the patients, so they need to have that second dose in order to help people become immune. Whereas with Johnson & Johnson, this DNA being more stable only requires this one dose. So again, logistically and in terms of administration, the DNA and the adenovirus vector is a much more reasonable approach to this. And so what happens once you get that injection from a Johnson & Johnson vaccine is basically you're going to get a bunch of these adenoviruses that are going to go into your bloodstream, and from there they're going to infect these host cells. So any one of your somatic cells in your body is basically going to have this adenovirus latch onto it, just like the coronavirus would latch onto it. And then what's going to happen is you're going to have this fusion event where basically this adenovirus is going to incorporate itself into a host cell. Inside of this cell, it's going to make its way into the nucleus of the cell where you're storing your DNA. And so it's going to inject its DNA here. And inside of that nucleus, the DNA for this adenovirus is going to get transcribed into RNA, specifically mRNA, that will then leave the nucleus, enter the cytoplasm, and from there, that mRNA is going to meet up with a ribosome, and that ribosome is going to actually translate this genetic material into a protein, specifically the spike protein, because that is how this Pfizer, all these vaccines are doing the same thing, but in this case, when we're looking at the Johnson & Johnson vaccine, this RNA inside of the host cell, this vaccinated host cell, is going to now produce these spike proteins. And so over time, we're going to produce a bunch of these spike proteins inside of a host cell, and as these spike proteins accumulate, basically the cell's functions, and it's basically diverting all of its resources into producing these spike proteins, which really don't help the cell survive, and it's actually going to end up killing the cell. But in addition to that, what it's going to do is these cells, these somatic cells, have this thing called a major histocompatibility complex, a MHC complex, where they basically will post on the outside of them what types of proteins are producing on the inside of them. And this is going to be important for helping your body's immune system down the road, but basically what's happening to this vaccinated cell that was infected by this engineered adenovirus from Johnson & Johnson is that it is posting these proteins on the outside of it that will help the immune system recognize this particular spike protein in the future if it ever sees it again, like when it sees a real coronavirus, which is how you become immune to it in the first place. And so what we have here is a picture of what is happening as this cell is basically dying, is that these cells basically burst, their membranes become permeable, they release everything to the outside world. All those spike proteins they had been making are now going to be released, and you're going to get these things called antigen presenting cells whose job is to basically walk around and grab stuff that they see that they think looks weird. So they're going to grab one of those spike proteins that's just floating around. They're going to do this thing where they basically degrade it using proteasomes, and they're going to display parts of this virus on the outsides of them. And when they do this, they're going to display this little part of the spike protein that they found to something called a helper T cell. And these helper T cells are going to go on and try to find a B cell to go stimulate, and specifically only B cells that have affinity for this particular protein fragment that they saw that they also got activated by. And so when a B cell sees this protein fragment, this B cell is going to do something where it basically is going to start cranking out as many antibodies as it possibly can. It's going to turn into this thing called a plasma cell. And so this plasma cell is just churning out tons of these antibodies that have a high affinity and a high specificity for this protein of interest. In this case, that protein of interest is this spike protein that you got from that helper T cell that came from the antigen presenting cell that found it from the vaccinated cell that had produced a bunch of those spike proteins that came from the adenovirus that was engineered by Johnson & Johnson upstream of it. So that's a sequence of events here, is that basically we've now got helper T cells as well as B cells that are cranking out all these factors that your immune system is already naturally doing, but now you've got a bunch of specialization specifically for the coronavirus itself. And so what's going to happen is that if you came into contact with the real coronavirus, because that coronavirus has that spike protein that Johnson & Johnson vaccine also had inside of its DNA, that antibody that was being produced by all those plasma cells, as well as those T helper cells, are going to be able to recognize that virus immediately. And antibodies, they're going to be doing two things. Number one, they're going to stick onto all these spike proteins on the outside of this virus, and by doing so, it is going to result in that virus being physically unable to infect anything, because basically it's just weighed down by all these antibodies that are blocking it sterically from able to actually use their spike proteins to grab onto a host cell. But in addition to that, when something has an antibody stuck onto it, if a macrophage or some other immune cell is walking around and it sees some other foreign object with an antibody on it, it knows that that foreign object is a bad guy. And so basically it's helping our immune system recognize that this virus is indeed a virus and it needs to be destroyed. So not only are we preventing this virus from infecting cells, but we're also able to now effectively recognize it and take it out. So that is the key takeaway here from how the Johnson & Johnson vaccine works. It's using that DNA that's much more stable than what we're seeing in the Moderna and Pfizer vaccines. And hopefully this is another very useful tool in our toolbox for how we're going to begin to control this pandemic. Hope you guys found this useful. Let me know if you have any questions, and take care. And also, please make sure to stay safe and practice social distancing and listen to what all these health officials are telling us. Thank you so much for watching. Take care. See you next time.